Hello, my name is Martin Horvath, and I'll be presenting this poster. You can also see the poster in real life in the second session. It's poster on Tuesday, poster number 16. And the title is Interbacterial Warfare in Plant Colonization. I'm a structural biologist, and I'm trying to understand how uh, tailocin, tailocins are targeted to specific bacteria. And we believe that to understand that, we have to understand the recognition of glycan receptors by the talus tail fibers. I'm part of a team at the University of Utah School of Biological Sciences. The lead scientist is Talia Karasov, and she has a talented graduate student, Talia Backman, and I'm assisted by two undergraduate students, Lauren Wynn and Hennesses. What are talusins? In simple cartoon terms, talusins are weapons that bacteria use to engage in mortal combat. And they provide some type of competitive advantage for pathogens that are uh, colonizing plants. Uh, the plant is a competitive space, and to win this battle, pathogenic plant microbes deploy molecular killing machines that resemble the tails of bacteriophages. And that's why we call them talusins. But even though they look like bacteriophages, or at least the tails of bacteriophages, they are not the same thing as bacteriophages. And the talusins have evolved for a different purpose. They do not replicate on their own. They're encoded by a cassette in, in, in the bacteria. Uh, but they do not replicate uh, autonomously. And they also do not carry a nucleic acid payload. And also different from bacteriophages, they kill target bacteria on contact. And they do this by deploying a spike which penetrates through the membrane to depolarize the victim and therefore dissipate the proton gradient. The key for killing specificity is located in these tail fibers at the very tips. There's a portion of the tail fiber protein that is looking for the receptor, a glycan receptor on the surface of the target bacteria. So to understand killing specificity, uh, one approach is to examine the structures of the tail fiber proteins and also the glycan receptors. And on the left here, we see four examples of tail fiber proteins. Their structures were predicted by CoLabFold. These are trimeric uh, fibrous proteins. They coil around, they're entwined around each other, the three subunits. And here at the C terminus is the portion that is looking for the glycan receptors. And interestingly, it's very difficult to predict which bacteria are targeted and which bacteria are resistant to a particular talusin, but the length of the tail fiber protein does correlate with killing specificity. And there are four main haplotypes for these tail fiber proteins that come from naturally occurring Pseudomonas viridiflava strains. This C-terminal portion is also the part that is most rapidly evolving, probably to keep up with changes in the glycan receptor on the surface of competing bacteria. Our collaborators at the University of Georgia have done some compositional analysis for bacteria that are sensitive and resistant to a particular talusin. And they found that a large substantial O antigen correlated with sensitivity, and this lipopolysaccharide structure was enriched in the carbohydrate rhamnose. So rhamnose may be a key component of the glycan receptor targeted by talisins. And interestingly, resistance does not emerge easily for these pathogenic strains, probably because there's a trade-off. If they lose the ability to build rhamnose into the O antigen, or they stop building an O antigen, they also are no longer pathogenic. So rhamnose, or this glycan receptor that is targeted by the talisins, may also be necessary for evading the plant immune system, or some other otherwise necessary for plant pathogenicity. And what I've done here is I've taken the predicted structure of one of these tail fiber proteins and then docked the chemical structure of L-rhamnose in both of its stereoisomers, the alpha and beta anomers. And these ligands, they land at particular places, these pockets on the surface of the tail fiber protein. So these appear to be constructed in a way that can accommodate rhamnose, or the rhamnose sugar group. And on the right-hand side, I'm comparing the affinity for different classes of proteins. TFB X-ray, this is a collection of proteins that are found in the protein data bank and that are tail fiber proteins, either from bacteriophage or from other types of talisins. The enzyme class, this is where I found the rhamnose ligands. These are enzymes that are interacting with rhamnose either as a substrate or as a product. These are carbohydrate modifying enzymes. And then in the middle are the predicted structures from these Pseudomonas verticillata talisin tail fibers in the 4 different lengths defined by how many amino acid residues are in the tail fiber. And interestingly, all of the docking outcomes were favorable, with a negative free energy of binding. And at least in a few examples, the talisin tail fibers from these Pseudomonas verticillata pathogens, they are binding to rhamnose with affinity that is even better than as seen for X-ray structures of other tail fiber proteins, or for enzymes that have evolved to act on rhamnose. To complement this this computer modeling approach, we're also trying to express tail fiber proteins, where we have expression systems that producing the tail fiber protein, even in the natural bacteria, requires a chaperone protein. And here in this heterologous protein expression system, we can express the chaperone protein, and we can co-express the chaperone protein with the tail fiber protein. I'd like to thank people in my lab, Lauren Wynn and NSS, who are co-authors on this poster. And also they've been joined by rotation students, Paige Dillon and Lydia Dresler, and also another undergraduate student, Alexa Toms. And then I also want to thank my collaborators and co-authors, Talia Karasov, Talia Backman, and then also the people at who are at other universities who are also helping us. Hernan Barbano is an evolutionary biologist who's been tracking these talisins and their tail fiber proteins across the last 200 years by looking at the metagenomes from herbarium samples, and then the carbohydrate analysis was done by Artur Izinski together with Parastu Azadi, who's here at this meeting. Here's those collaborators again, and I'd like to call attention to some articles you can read about many of these findings as described in a recent research article in Science, and then the whole subject of what are tail lizards doing in wild populations of Pseudomonas. You can read about that. Both of these articles are by Talia Backman this year, and the review article is in Trends in Microbiology. And this work was supported by funding from the NSF and from the U.S. Department of Agriculture. Thank you for listening.